name is Mira. I'm with the Tufts University Center for Cellular Agriculture, which we call TUCCA for short. And we are an academic research center focused on all things cell ag, so cultivated meat plus other products, which is what we're going to talk about today. So I'm very excited to welcome Stephen Rio from California Cultured, and I'll let you take it away.
Great. Thanks so much, Mira. And also special thanks to the Tufts University Center for Cellular Agriculture. I think that's good work being done there and really pushing the industry forward. I'm joined here with Rio. Rio's the lead scientist and associate director at California Cultured, and he and I are going to be tag teaming this presentation. Rio's been with the company 2.5 years. I've been with the company about 6 months or less than 6 months. And then previous to that, I was with Upside Foods. So I'm able to give a little bit of perspective on both the cultivated meat side as well as cultivated cocoa. So Rio and I landed on a title for our talk that is a little bit of, I hope, thought-provoking. What we're trying to demonstrate here, or maybe get you to think about, is how can new cellular agriculture technologies like plant cell culture help move the entire industry forward and take away some of the headwinds that have been really buffeting the cultured meat sector of this industry, and kind of eliminating some of the blowback from the cultured meat space that is maybe impacting the larger industry. So as we go through the presentation, hopefully that becomes a little bit more clear and focuses on how plant cell culture may help the entire agriculture industry. So to begin with, it's not advancing. Should have practiced this. There we are. So a little bit about California California Cultured, excuse me. We're a small startup out here in Sacramento, California, and we are developing product lines around chocolate and coffee. And the whole idea is to take away the negative externalities that come about with the conventional farming of chocolate and coffee. That being namely destruction of rainforests, as well as unethical labor practices in both of those industries. And ultimately, we're working on developing processes that will be sustainable and an integral part of our food system going forward. We'll be talking mostly about chocolate in this presentation because that is our lead product, and hopefully on the market by the end of this year. So without further ado, kind of get into some of the science and the strategy around cocoa powder. So we consider ourselves a cocoa powder company. And because we're building this traditional or conventional product with novel manufacturing processes that are very akin to cultivated meat. And that is exactly as demonstrated here. We collect cells from the cacao pod, select and screen those cells for different attributes that we want in our product. We scale that up and produce it, and then dry it down, and that is essentially our product. Now, in the selection and screening stage, of course, we're looking for different attributes, much like you would look for different attributes in animal cell culture. Part of that is around what are the product drivers for us being interested in cocoa. And if you've been paying attention to the news in commodities over the last couple of years, you would maybe have known that your chocolate prices are increasing. And in fact, it was last Halloween, wasn't it, that there was a lot of news around more people going after the sugary treats rather than the chocolate treats, just because of the cost of those treats. So what we've been experiencing across the globe is decreasing supply and increasing demand for cocoa. And that's been really highlighted in commodity news, as shown there. Cocoa has gained almost 185% in 2024. And we don't anticipate these prices to decrease or supply to increase and demand to decrease. In fact, we expect the opposite. Increases or continued problems with climate change, weather event, governmental policies, and of course, disease in cocoa farming will continue to constrain the supply of cacao. On the other hand, we'll have a growing middle-class population across the globe, and they are looking to purchase more richer foods, much like meat and chocolate. So we anticipate the increasing demand for chocolate to continue. And then the other part of our company is really about the health of chocolate. So I think everyone knows that dark chocolate is good for you. And what we're producing here is something that is extrapolating that to an extreme. So the cocoa powder that we're producing, we've identified a cell line that produces high concentrations of flavanols. And those are the bioactive compounds that are responsible for the clinical benefits of dark chocolate. And just highlighting here one of the studies called the Cosmos trial, where they were able to demonstrate that cocoa extract supplementation greatly reduced cardiovascular death in the participants in that trial. So it's not only cardiovascular, but there's also benefits to your immune system and inflammation, as well as your microbiome, and also stroke reduction. So these flavanols, these bioactive compounds, are really beneficial to everyone who consumes them. And then so we are here at California Cultured, basically trying to create a bio-better chocolate that is actually really good for you by increasing the concentrations of flavanols that we can transfer to our customers. The other healthy aspect of our product is that it comes free of heavy metals, pesticides, and antibiotics. So heavy metals are particularly concerning in chocolate because the cacao plant basically bioaccumulates heavy metals into the pods. And that then is transferred to the consumer. And it's something I think there was in the literature, something about 50% of the chocolate that's on the market have concerning levels of heavy metals, particularly lead and cadmium, in their products. So this is a real concern that the FDA is paying closer attention to, and they have an initiative called Closer to Zero in order to get lead levels down or out of our food system and down to at least as close as possible to zero in our foods. So we see what we're doing here is, instrumental to not only our benefiting health, but also leading along with the regulatory authorities in this effort to create healthier food systems. And this is important because, coffee and chocolate represent a huge market. There are nearly 2 billion people across the globe that daily consume either cocoa or coffee. And it's perceived as a premium product. So people really, aspire to consume it. And that is good for us as a startup company because we can demand a higher price point as we start to sell our product because we're healthier, more sustainable, et cetera. And as we reduce and optimize, reduce our COGS and optimize our processes, we'll get closer to price parity. But in the beginning, we can charge a price premium to keep us moving forward. The last thing that I think is really important from the food security perspective is essentially onshoring cocoa production to the entire globe. So cocoa production is limited to only the equatorial countries where the climate and the humidity, all of the factors are right for cacao farming. And what we're doing here at California Cultured is basically being able to remove the plant and only use the cells. We're able to then essentially build a biomanufacturing facility anywhere in the globe and co-localize it right next to the chocolatiers or whoever would be our customers. And I think this is going to be a big boon going forward as we think about onshoring and bringing jobs back to locales. More on a strategic and getting into the technical comparison between plant cell culture and animal cell culture and directly kind of comparing cultured cocoa and cultivated meat. First thing we want to think about is, of course, safety. And giving plant cell culture high marks in the safety realm because there are no known animal or human pathogens that can be transmitted through plant cells to the consumer. So that was, of course, a big concern in the beginnings of biopharmaceuticals, which are developed all the technology that is now being used in cultured meat. And the transmission of pathogens through the processing was a concern. And we've now demonstrated that we've got mitigation plans in place and techniques available to reduce the risk of zoonoses or pathogen transfer through animal cell culture. But there's always still going to be a risk, and that risk is not present in plant cells. The next thing is looking at scalability. Animal cell culture, typically in biopharma, goes up to about 20,000 liters. And I don't know of any cultivated meat companies that have gone beyond that. And to really hit the cost of goods for cultured meat, you have to rely on economies of scales and going to very large scale production beyond the 20,000 liter. On the flip side, in the plant cell culture space, we've got demonstrated scalability of up to, if not beyond, 75,000 liter capacity. So plant cell culture seems to be much more scalable than animal cell culture. If we move on to cost effectiveness, I think everyone on this call probably appreciates that the operating expenses for any kind of cell culture is going to be driven by the media cost and then the cleaning and sterilization cost. So animal cell culture still relies on clean in place, steam in place technology, whereas the plant cell culture, because plants make antimicrobial compounds in their biomass, it's reduced risk of contamination and therefore slightly better at the cost effectiveness because you won't have so many batches going down. I'd also like to say that the media cost for plant cell culture is going to be much different than animal cell culture, just because we have maybe a third of the number of components that we have in our media compared to an animal cell culture. So it's generally cheaper media. And then finally, I want to just talk about consumer acceptance, because I think this has gotten a lot of press recently over the last year, is people's willingness to try novel foods. And I'm relying on a report from 2023 by Gillico-Cologne and Yeager. I'm showing you here the synopsis of their data, which is a survey of about 2,500 people across 3 different countries, asking them for their willingness to try novel food products. And they rank them from 6 to 1, 6 being the highest likelihood to try. And you can think of that as likelihood to try, and also then an indication of repeat purchase. So what is it? Urban farmed vegetables, very likely to try. Same with modified packaging vegetables, very likely to try. But if you look at the bottom, it's cultivated or cell cultured fish, cell cultured meat, and then meat that's been supplemented with 10% insect meal. So we have this in cultivated meat space, this ick factor that we have to overcome to get consumers to accept cultivated meat as a safe and effective way to deliver nutritious proteins to people. And that's going to be a hurdle that will require some work to get over. And I think this also pushes or demonstrates that plant-based, like this plant-based 100% milk, is much more likely to be accepted by consumers and repeat customers. So I'm going to argue that by using plant cell culture, we can actually push the needle on consumer acceptance of these type of novel food products in order to help the entire cellular agriculture industry in moving towards becoming much more mainstream, both in plant cell culture as well as animal cell culture products. So if we look at what we're talking about here, is cultured meat is very much, and I'm sure everyone's very familiar with this, the idea of taking animal cells from domestically produced farm animals and then immortalizing them, banking them, and then expanding those either in suspension or adherent cell culture to a point where you can produce enough biomass that you can dewater it. And then that biomass is then further formulated either through printing or scaffolds, differentiation, or mixing with plant-based protein to formulate it into a surrogate for meat, that is a cultured meat product. Here at California Cultured, we're doing exactly the same sort of thing, but it's a little bit simpler. And that's shown here. So, and this is where Rio is going to get into and explain some of the details around this. But on the high level, we have essentially unit operations that are very analogous to animal cell culture or cultivated meat, except a lot less downstream processing. So let me explain. So we take cells from the cocoa bean, generate cell lines in the lab that we can control and then grow into a suspension culture that is then scalable up to, 100,000 liters. After that, our downstream processing is very simple. We essentially dewater and then dry. And that is a picture of our product, which looks very much like conventional cocoa powder. So at that point, I'm going to turn it over to Rio, and we'll learn a little bit about plant sciences.
Yeah, thanks, Steve. So we thought it would be useful to kind of highlight some of the differences in basic plant biology. There are a lot of similarities. These are complex multicellular organisms. They have multiple tissue types that are spatially and temporally regulated. They contain active centers of cell division, analogous to stem cell niches within animals. However, in plants, we call them meristems. And the key difference here is that in animals, you have a multitude of distinct stem cell populations, each one genetically pre-programmed to produce a very specific set of subsequent cell types or tissues. So that if you take a biopsy, you have to find the exact stem cell that you're interested in and then maintain it as one population that can produce one type of tissue. Whereas in plants, we have three broad meristems, which is what we call our stem cell niches. So these are sites of active cell division. They maintain an active population undergoing mitosis, and these are typically undifferentiated self-renewing stem cell populations. However, unlike in animals, we have much simpler organization of these meristems. So we have a shoot apical meristem, which is at the growing tip of a plant. So this is responsible for producing new shoots and new leaves. And then at the other end, we have root apical meristems, which are responsible for producing new roots. And so together, these constitute what we call primary growth, which you could think of as lengthening of the plant during development. There is also what we call secondary growth, which is derived from a meristem population that resides in the stem of the plant. So this is responsible for producing new vascular elements in response to environmental cues. And it's also what's responsible for thickening of a plant stem as it grows. And so if we look at a tree from season to season, the main trunk of a tree is not growing upwards, it's growing outwards through this lateral meristem activity. And so if we zoom in on a shoot apical meristem, what we see is that the cap of a plant, the part that we see, is called a bud. And so within that bud, we have preformed leaf primordia that are essentially contain all the cells within the leaf that will that have already gone through division. And the meristem itself is restricted to this black cap at the apex of that dome. And so if you notice is that the cells in the more developed tissues around it are large enough to see. And so they've undergone cell expansion and some level of differentiation into their final phenotype. Whereas the cells within this apical meristem are very small. They typically have dense cytoplasm, and they're maintained in an undifferentiated state. And so we have developed what's called a tunica corpus model of the meristem. And so that there are essentially 3 cell types within this meristem divided into layers 1, 2, and 3. Layers 1 and 2 divide symmetrically in a lateral fashion, such that division happening at the center of the apex pushes cells outward and into the developing leaf primordia that you see above it. Whereas, and so these become the epiderm and the subepiderm of a leaf, whereas the tissue beneath it is called L3, divides both laterally and vertically to create what's called a ground layer. And so this is responsible for the bulk of material that you see in a tree trunk, and it's also what develops into vascular tissues. And so in plants, we have xylem vessels, which are responsible for transporting water, and we have phloem vessels, which are responsible for transporting nutrients and sugar throughout the plant. And so where this really diverges from the animal model is that stem cell identity in plants is defined by their cellular environment and not strictly by their origin. And so what maintains these stem cell niches is an interplay between 2 genes. We have WUSCHEL, which is a stem cell promoting transcription factor, and clavata3, which is a differentiation promoting peptide. And so these create a feedback loop that regulates their expression to a very defined spatial niche within that apical meristem. And what's really unique and where we can see the differences is you can actually ablate or destroy this active dividing center, and new active dividing centers will spontaneously emerge from existing tissues. And so there is this concept within plant biology that spatial organization is sufficient to drive cell differentiation to create new niches of division, basically. And so the effect of this is that we have totipotency and plasticity in plant cell culture that does not exist within the animal cell culture space. And so by totipotency, we mean that a single somatic cell can regenerate into a fully developed adult organism without undergoing meiosis and gamete fusion. And plasticity refers to this idea that the cells within that actively dividing meristem can go on to assume many different final phenotypes depending on their local environment. So there is not strict developmental pathways as there is in an animal system. So when we get into cell culturing plants, we start with what's called an explant. This is analogous to a biopsy from an animal. See it shown here in the center. And from a single explant, you have many different developmental paths that can be achieved by modifying the environment of that explant. And so plants can directly form new shoots and roots through what's called direct organogenesis. And so this is new tissues deriving directly from differentiated explant material, or we can direct the growth into an or we can push it into what we call callus. And so callus in plant cell culture is a unorganized, dedifferentiated cell mass that grows through cell division. And so once you have this sort of mass of plant cells, because our meristems can form spontaneously through these feedback loops, we can push this dedifferentiated mass of cells into indirect organogenesis, as what we're seeing on the left. So we can produce new shoot or root apical meristems directly from this tissue, from this cell mass. We can also take a callus culture and move it directly into liquid media to produce a suspension culture. And so unlike animal cell culture, plants spontaneously grow and divide in a suspension environment, and there's no need for scaffolding or reprogramming cells to grow in a suspension environment. And because plants have a cell wall, one of the main differentiating features, this is a barrier to genetic modification. And so it prevents entry of external materials into the cell. And so one of the techniques involved is called a protoplast culture. So you can remove cell walls from a plant, and you get a protoplast that is much easier to deliver foreign DNA for genetic modification, or CRISPR reagents for gene editing. This is also a path for creating novel hybrids. You can induce two distinct cultivars or even species of plants to fuse and form a novel hybrid. And then finally, plants have what are known as somatic embryogenesis pathway. And so similar to the animal world, plant embryos are the product of gamete fusion. They undergo meiosis and gamete fusion to create a fertilized egg cell that has a predetermined developmental path to create an embryo that exists within a seed. And so unique to plants is that we can coax a somatic cell into following this morphologically defined pathway of an embryo that we call a somatic embryo. And so what this gives us is many different pathways and many different types of cell lines that can be derived from a single plant.
All based on the environment that we're providing them.
Yes, based on the environment that you are pushing them through, and not strictly dependent on the particular cell that you isolated from an organism.
Right.
So plant cell culture is a fairly old field. It's been around since the 1950s. Traditionally, these have been used as tools to facilitate traditional agriculture. And so one of the main primary starting points of this was clonal propagation. We've seen that they can spontaneously form new shoot meristems. And so this lets you take a single adult plant and clonally multiply it to produce as many progeny as you want, all with the exact same genetics. And so this has been heavily used, particularly in crops where seed is either not available or genetic diversity is too high such that you cannot produce genetically uniform seed populations. These also provide tools to enhance traditional breeding. So breeding, we are trying to cross distinct members within a species to isolate and recombine phenotypes to get sort of this improved plant out in the field. And so these techniques can be used to produce novel hybrids as well as accelerate the process of breeding. And then in the last 30 years or so, people have been using tissue culture as a tool, or we'll call it the bottleneck, for genetic modification. And so if we want to transform a cell and regrow it into a new plant with genetic modifications, it has to pass through a cell culture pathway. But the commonality between all of these is that we started with an adult plant and we ended with a new adult plant that fed into a traditional agricultural setting. And so starting in the 80s, actually, plants were some of the very first cellular agriculture ventures that came about. And this comes from a long-term understanding that plants produce bioactive compounds. So these are useful molecules that elicit a response when you consume them in some way. And so we've long known that plants are medicine. Aspirin was derived from willow bark before we figured out how to synthesize it. Antimicrobials, anti-inflammatory agents. We're now looking at dyes and fragrances and supplements. So we've long known that plants produce valuable metabolites. And in the 80s, people started to utilize cell culture techniques to molecular farm metabolites in a controlled environment. And so there's lots of advantages to this. As we've seen in our product, you can achieve much higher concentrations of specific metabolites through cell culture techniques. And probably one of the biggest wins that has come out of this is a compound called paclitaxel. And so this is a highly potent and efficacious anti-cancer drug that's found within taxus species. So these are very efficacious, but they are produced at very low quantities in a very slow growing tree species. And so this is one of the real wins to come out of this molecular farming approach for plant cell culture. And so we wanted to highlight just a couple. So similar to cell line development for animal agriculture, you start with an explant. You need to induce, you send it down this pathway of cell de-differentiation and then organogenesis into different tissues, or we maintain them as a suspension culture of de-differentiated cells. But no matter what pathway you go down, you have to select the cell line of interest. And so this is often based on growth rate and metabolite concentration. You need to optimize culture conditions. You can elicit secondary metabolism. So in plants, we can control primary growth and secondary metabolism through the use of elicitors. And you can also increase titers through techniques such as precursor feedings or immobilization. But once you have a selected cell line in an optimized growing environment, you can go into a bioreactor, scale this up, and then harvest your compound of interest. And so here we're seeing paclitaxel molecule coming out of a bioreactor. So this is one of the more scaled approaches. So Steve, in his introduction, mentioned that plant cell culture has scaled up to 75,000 liters. This is coming from a company called Phyton Biotech. They're based out of Germany, and they're now expanding into Canada. So they have taken a very traditional approach to plant cell agriculture. They're using stainless steel impeller mixed bioreactors. They have a cascade of ever increasing sizes, finishing out with the 75,000 liter bioreactor. And so what we're seeing here, this is from their website, is just the tip of the 75,000 liter vessel and a mezzanine essentially, so that you can access the upper parts of the bioreactor with the body extending down into it. And so this is very analogous to traditional biopharma approaches. They scale. They achieve product recovery from broth, and they can achieve up to 800 kilograms paclitaxel production per run. So this is a scale up approach, build ever larger bioreactors. Plant cell culture is also feasible with a scale out approach. And so here we're highlighting a company called Protalix Therapeutics. They're based out of Israel. And so they make recombinant enzymes in plant cell culture systems using their proprietary bag system that you see here. And so rather than one target pharma drug, they are producing a range of drugs targeting multiple rare diseases. And so for them, a scale out approach with modular production makes sense to fit their needs. And so what we see is that there's many paths to grow plant cells. Each one provides a different cost of goods. The trick is finding a path that will get you to a price point that fits you in the market, basically. And so we saw Phyton Biotech is very successful with a pharma-like infrastructure, but they're making a pharma product and they're getting pharma prices for it. And so one of our basic underlying principles is that this existing infrastructure works, but it will never deliver us the cost of goods that we need to produce a commodity product at commodity prices. And so that is what we see as the real problem for us to solve, is not cell culture, but how do we do it at a price point that produces the product we want and gets it to the consumer.
Yep. So on that point, then, I think what Rio really kind of highlighted there is that these plant cells can be hugely flexible. And that flexibility brings in a lot of opportunities for new technology, as well as then innovation that will allow us to bring the COGS down and get these market or products into the market much faster. So just to reiterate, Rio talked about all the multiple different modes of growth, the cali, the hairy root, cell suspension, et cetera. But also then there are multiple production systems. So was it Phyton was using airlift, and then the first one was stir tank reactors. But then there's also bubblers, misters, immersion bioreactors that you can use in cultivating plant cells. And I need to also emphasize here that we have a simpler media and feeds. Our process for harvest and downstream processing is much more streamlined. Simpler environmental controls, basically for our plant cells. We only control temperature around ambient and monitor the DO. As Rio alluded to as well, we can use elicitors to go from the growth phase to induce them to produce those metabolites that we're really interested in. And then because the cells tend to aggregate and are larger, that gives us windows into ways that we can do continuous manufacturing that maybe isn't possible with mammalian cells or animal cells. And then the last thing about the flexibility in plant cell culture is that because we're controlling the environment, which is paramount for these cells, we can envision taking a single biomanufacturing process and turning it into multiple different products, be it a food product or a supplements, or extracting the bioactives completely and just having pure bioactives. So all of these things kind of lend itself to the idea that plant cell culture may be actually leapfrogging animal cell culture and getting into the forefront and then helping us deliver the promises on cellular agriculture. It is not all rainbows and unicorns. Plant cell culture is still very challenging. There are challenges that we have to deal with every new cell line that we develop. First and foremost is that they're slow. This is not a microbial 15-minute doubling time. Typically, our cell lines, when we start out, double time is about 3 to 4 days. And the mitigations there is really around cell selection, adaptation, and then continuously controlling that environment to optimize the process and the cell growth. The other interesting thing about plant cell culture is because of the cell wall, cryopreservation doesn't work as well as it does in animal cells. So for long-term storage, we have to develop cell line specific conditions for cryopreservation or come up with alternative methods to have these seed cultures preserved and stored over a long period of time. And then getting into the phenotypic plasticity and variability, yes. Plant cells, much like animal cells, if you don't control the cell line identity, it will diverge, and cell line stability is a concern for both animals and plant cells. But we believe in plant cell culture that controlling that environment is a lot easier. And then finally, the last challenge I'd just bring up is, as Rio was mentioning, the plant cell wall really does prohibit from introducing new DNA elements into the genome of the plant. And so, our approach has been going with a non-GMO or not doing genetic modification, but then just screening and selecting for phenotypes that we want. But there are techniques for introducing genetic modifications like through protoplast technology. So while there are challenges, a lot of them have been mitigated or have alternatives, and really comes back to the idea that plant cell culture has a lot of advantages that we don't see in the animal cell culture. And in conclusion, what I'd like to kind of point out is that I think regardless of, if you're talking about cultivated cocoa versus cultured meat, through life cycle analysis, we've demonstrated that biotechnology techniques can actually transform crops into food very efficiently. And that is traditionally been done through feeding crops to an animal to produce meat, for example. But in the cultured meat space, at scale production, really cost-effective products, and in the market demand, yet is elusive. And what we're arguing here at California Cultured is if we can take a plant cell and turn it into a plant product, we may be able to overcome a lot of that consumer reluctance or willingness to try those novel foods and demonstrate that cellular agriculture is going to be the future and an important part of our food systems going forward. And we can do that with plant cell culture because we've demonstrated at scale production. We can create cost-effective products in very large markets so we can make money doing it. So healthy cocoa and chocolate is going to be our first product on the market, but I want to leave you with the idea that we're going to disrupt the coffee market as well. So coffee is following the same trend lines as cocoa, hit by climate change issues, and the prices are skyrocketing. So we're anticipating that with recent advances in our lab that look like some really good cell lines, that we'll be developing new coffee products very soon. So with that, I'll stop and really thank you for your attention and open up the floor to questions.
Thank you. We have some questions in the Q&A, which I can ask. Are you able to see them as well?
We have the chat open.
But not the Q&A.
Okay, it's down at the bottom, but I can just start asking from there.
Right. Yeah.
Which one should I start with? From John, I've heard that bacteria can be preserved via drying because of their cell walls. Have researchers looked at this option for preserving plant cells?
That's an interesting one. I mean, when I think of dried microbials, I think of spores. So those are quite different. But Rio, do you have anything to add there?
Yeah, the only way to preserve a plant through drying is with seed.
Mm-hmm.
Whereas actively growing, if you want to maintain a cell line, we cannot dry it down and rehydrate it the way we would with a bacterial culture.
Yeah. Thanks, John. That was a good question.
And then when you're looking into different scale up versus scale out configurations for your process, are you considering other factors besides cost, like environmental footprint, facility size, or resource usage? And do you have thoughts on what the better method is for scale up versus scale out?
So yeah, we're exploring all of it, and we have two-pronged approach, I would say. For what we're able to do currently, we're working on getting up to relevant scales. We're hoping to have within the next couple of months here a 2,000 liter at our R&D facility, and then we're thinking about how to build that into a food grade facility thereafter. But we are also in conversations with CDMOs to do the scale up as well. What we want to do is essentially convince our customers that we can produce consistent supplies that they would need. And scaling up and demonstrating that economies of scale seems to be really important to consumers or our customers, which are B2B.
Yeah, speaking of economies of scale, I think everyone always has this question. What's the likelihood that you're going to be able to get to cost parity with commodity chocolate, and when would that be?
So that's a great question, and I anticipate, as I said in the talk, that conventional cocoa prices are going to continue skyrocketing because of course, the arable land that's farmed for cacao, and also the increasing demand. So we are targeting, from our process economics, and I don't know how much I can share here. We're looking at reaching price parity within the next couple of years.
Okay.
Yep.
And then do you leverage AI or machine learning in the production pipeline or to help with the scale up?
Wow, I would love it. But with every R&D organization that I've been associated with, there's always been the problem of where do you get all of the data that you use to train and use AI? And many R&D organizations just are not structured in a digitization mentality to be able to really capture the value of AI, particularly in a biomanufacturing or an R&D stage.
Regarding the regulatory landscape, could you please explain how the process of obtaining a no questions letter for cultivated chocolate or coffee differs from the process for cultivated meat?
So it's largely the same. I mean, it's a GRAS process, generally regarded as safe. And there are multiple paths you can go down with that GRAS process. You can do self-GRAS or just the normal GRAS process. And how it is different, I do not know a lot of the details around that from cultivated meat. Rio, do you have any insight?
No, but similar process. We want to convince the FDA that what we produce is safe and reproducible, and known, understood. So we fully characterize the profile of what we make and show that it's consistent and reproducible across runs.
Right.
We have a few questions about flavor. Yeah, does it actually taste like chocolate? One person, anonymous attendee, asks, and Taiwan asks, have you investigated the flavor? Like, are you able to control flavor by changing growth media?
Yes. So first of all, I'll say yes. Our chocolate tastes great. I'm paid to say that. No, I'm kidding.
Yeah.
No, I can honestly say that the cocoa powder that we use, and you have to remember that chocolate is made from sugar, cocoa butter, and cocoa powder. And by adding our high flavanol cocoa powder to the preparation of chocolate, results in very good chocolate. It tastes like how was it described? It tastes like a 75% cacao chocolate. No, I'm sorry, I've got that wrong. It has the taste of a 55, but it really has a flavanol content of a 75, something like that.
Sure.
I don't know about
the interesting thing about chocolate is that all of the flavors that we associate as being chocolatey come about through downstream processing.
Mhm.
So a raw cocoa bean from the tree has almost nothing in it that would remind you of chocolate. And so the flavors of chocolate are produced through fermentation and roasting and subsequent downstream processing. And so when we push our cell biomass through a similar process, we achieve chocolatey flavors. But it's largely a result of downstream processing and food science as opposed to cell culture techniques.
And how much of that downstream processing would California Culture do versus your B2B partners?
Well, depends on the product. If we're selling cocoa powder for a different purpose than maybe cocoa or chocolate, then we would just be selling the dried cocoa powder. But if we were targeting maybe a chocolatier that wanted us to do the fermentation, we could do that.
Cool.
Yeah.
If you were working on a non-food product derived from plants, what product would you want to produce, or do you think makes sense to produce?
Non-food product. besides pharmaceuticals.
Yeah, I think probably besides pharmaceuticals.
Yeah. Well, I mean, there's so many options. It's hard to pick out what would be best to go after. You have an answer?
I mean, I think the way we came to our choices was through a market analysis in a
Right.
lot of ways. So
Yep.
you want something that, you want a product that has demand
Mm-hmm.
and has some sort of restrictions on its current production, and then has a premium for quality control that you can bring in a bioreactor. But what that is, I don't know.
Great question.
What yields are you able to accomplish per liter, and are you confident in being able to increase yield without genetic engineering?
Can we share that? Yeah, I think
I
that's
don't know.
very public knowledge. I mean, with plant cell culture, you can get up easily up into 30% packed cell volume, which is kind of unheard of, at least in my understanding in the animal cell culture space. So we can get yields that are
40% PCV.
yeah, up to 40% maybe.
Yeah.
And then I also think that with process optimization, intensification, taking learnings from biopharmaceutical processes, we can do much better than that. So we're right now working on a relatively non-optimized process that's producing very well. And we think there's a lot to gain yet from further process development.
Is virus contamination an issue for plant cell cultures?
So no, I mean, as I mentioned, there are no known zoonoses or viruses, human pathogens or animal pathogens that can be transmitted through plant cell culture. The one paper I was able to find on something similar, which is termed the word phytonoses, an analogy to a zoonoses, is pathogens from plants to humans. And that was simply mycoplasma. And that risk is everywhere. So no, I don't think there are viral concerns around plant cell culture.
There are plant viruses
Sure.
That impact plant growth.
Yep.
However, often the best way to eliminate a virus from a plant is to push it through cell culture. And so actively dividing cultures often will shed plant viruses, but it is something to keep in mind. You can have cryptic plant viruses that impact plant health.
Mm-hmm.
We have a couple more questions coming in. Are there methods being investigated to reduce the doubling time of plant cell culture, and how are cell banks in plant cell culture currently maintained?
You want to take that one?
Yeah, so you can definitely do cryopreservation. It is possible. As Steve mentioned, it's just line specific. So each one is kind of its own research process. But Phyton Biosciences, which makes paclitaxel, they claim to bank all of their lines in cryo.
Mm-hmm.
And I think the main barrier is just that it takes a longer time to bring them out of cryo and bring them up into production compared to a microbial system.
Or mammalian.
Yeah, or mammalian.
Yeah.
In terms of increasing doubling time, certainly in the plant world, increasing biomass and increasing yield is what we've been breeding for thousands of years. But it's not really the same thing as doubling time. And so I think that is very much open space. That is room for improvement, particularly in an academic setting. These are questions that no one has really had to ask before because there wasn't an industry relevant reason to ask those questions.
But we know it's possible. So there are model plant cell lines that double at about 24 hours.
Yeah.
So plants can do it. And what we have to do is, of course, for each cell line that we develop, figure out how to get them to produce faster as well, to increase their yields.
What are the biggest ongoing production costs for plant cell culture apart from the initial investment in equipment?
So it's basically employees and media costs. So that's going to be the biggest part. And then I'll say also, because we're selling a dried product, the drying is going to be another big expense. So just removing all of that water.
For cell lines, the questions keep coming in, but we'll go for a few more minutes. For cell line selection, do you analyze the cells through biomass production, metabolite generation? In addition, do you use flow cytometry or imaging or metabolite analysis for this?
Yeah, so metabolite yield and growth rates are the primary driver of cell selection. But cell morphology is something that we track very closely as well. We don't use flow cytometry, but it certainly is a path that could be taken for plant cells. If you can find a flow cytometer that will work with plant cells, it certainly is a way that you could segregate them into buckets.
Right.
And then are bioreactor designs more similar to microbial or animal cell culture designs, or neither, besides the size?
So yeah, as I mentioned, plant cells will grow in pretty much any type of bioreactor that you can put them in. We've had success with stirred tank reactors as well as airlift reactors. So what we're working on is basically trying to figure out how can we reduce the cost, as it was mentioned, the upfront cost of those capital investments for bioreactors when we need to scale up and scale out as much as we do to capture a little bit of this market share, and really looking at all alternatives for how to do that well.
And then I think this will be the final question, so you can let people have dinner. What is the biggest problem you face in your production pipeline or go-to-market strategy that you wish you had more time to work on?
Well, I mean, as with, I think every startup, your first product is one that you wish you had more time to work on. But generating revenue, getting it onto the market is paramount for success in the long run. So yeah, I think there are always going to be things that we'll want to work on, because we're academic and scientists. We're always curious on how to improve things. But with most startups, I think, you basically have to balance the needle with getting to the market, as well as then producing a good product that has consumer satisfaction. Anyone
Great.
add there?
No, I mean, like you said, in an ideal world, you'd have all the time to fully optimize a product, but
right.
there's no real such thing as optimized. It's
Exactly.
just good enough to get to market.
Yeah.
Any final words you want to share with the group? There are a lot of students on the Zoom right now, so any words of wisdom?
I would, really kind of think about what are the products that are missing in the world, and how can we use our technology that is developing today, and not be, be agnostic about the technology as you look at it. So, I'm very surprised that I'm working on plant cell culture to make cocoa after, maybe 20 years of cell culture experience in both biopharma and food space. So I would not tell anyone to discount technology immediately. So continue pushing the envelope on all of the technologies and see what works best.
Yeah, study plants. They're fascinating, and they're, compared to animal systems, are really uncharacterized. And so there is just a mountain of material that you could go into and find your own space in the plant world. So there may not be the money and the prestige as a biopharma space, but they are fascinating, and there's lots to learn still about them.
That is very true.
Great. Thank you so much for, sharing today. And thank you everybody for joining. This was recorded, so we'll edit the recording and then upload it to our YouTube channel later for folks who might have missed today. And with that, yeah, have a great evening, everybody. And thank you so much.